Καλησπέρα σας και καλώς ήρθατε. Dear colleagues, ladies and gentlemen, good evening and welcome to the 12th hybrid and final Zoom lecture of the 60th public lecture series of the Archaeological Research Unit of the University of Cyprus. It is my pleasure to introduce our today's speaker, Maria Rousou. Dr. Rousou is currently working as an archaeological officer on contract at the Department of Antiquities of the Deputy Ministry of Culture of the Republic of Cyprus. She completed her undergraduate studies at the University of Cyprus in 2016 and continued with her MA at the National Natural History Museum in Paris. Her PhD research was conducted in co-tutelle between the National Natural History Museum and the University of Cyprus under the joint supervision of Dr. Urania Kouka and Professor Margareta Tenberg. Maria's PhD thesis, entitled Exploitation of Plant Resources and Environmental Impact of the First Human Settlers in Cyprus, Anthropological and Carpological Approaches, defended in 2022, dealt with the questions of human-environment interactions and uses of plant resources and the impact of human activities on the environment during the pre-Pottery Neolithic in Cyprus. After completing her doctoral research, she continued as a postdoctoral researcher at the Malcolm Wiener Laboratory of the American School of Classical Studies at Athens, in collaboration with the University of Giselle in Algeria, on a multidisciplinary study related to the production of pistachia fixed oil in the Mediterranean. She has been involved in the sampling, flotation, study, and analysis of anthropological and archaeobotanical material from various Epipaleolithic, Neolithic, Bronze and Iron Age sites in Greece and in Cyprus. She has authored and co-authored articles published in peer-reviewed journals, in conference proceedings, and monographs of final publications of archaeological sites. She has also participated in numerous international scientific conferences in Cyprus and Europe. Her research interests include the reconstruction of the paleoenvironment and the study of human-environment interactions, the development of ethnobotanical studies, as well as the study of plant management practices and the uses of plant resources in the Eastern Mediterranean and the Aegean across time. Closing this introduction, I would like to kindly ask all our participants on Zoom to keep their cameras off and their microphones muted. Should you wish to address a question or comment to our speaker, feel free to use the chat button on Zoom, or alternatively, you may switch on your cameras and at the end of the presentation to address your question directly to the speaker by raising your hand and unmuting your microphone. On a final note, I would like to inform you that this is the last guest speaker in our public lecture series that I welcome to the podium in my capacity as Director of the Archaeological Research Unit, as my service expires on January the 15th, 2024. I must admit it has been a great joy and a wonderful experience to prepare and coordinate the program of the last 8 ARU series, especially since we started broadcasting on Zoom since September 2020. I would like to sincerely thank all my colleagues at the ARU for supporting this venture, for suggesting all those brilliant researchers and speakers from across the world to deliver a public lecture for the ARU, all the fantastic speakers in our last 8 series for their enthusiasm and hard work, and of course our technician Konstantinos and all our dedicated Zoom attendees for being with us on Monday evenings, supporting us, participating and interacting with our speakers. Last but not least, I would like to extend my best wishes to the ARU Deputy Director in January and to the new Director from September 2024 onwards for a most successful service. Thank you all very much. Dear Maria, welcome back to the ARU, and we very much look forward to hearing all about the contribution of botanical reference collection to archaeological studies. Thank you.
Hello, everyone. Dear professors, colleagues, friends, family, thank you very much for being here today. It's a great honor to come back to the Archaeological Research Unit as a speaker. Today I will talk about the reference collections that I created in the framework of my PhD research conducted in Paris and at the University of Cyprus. So I will not talk about the New Cyprus Museum, nor about the pistachia. This is one of my favorite topics, but I will present you the importance and the creation of reference collection that are used for the development of archaeobotanical and archaeological studies. I'm sure that all of you know the importance of archaeobotany and its contribution to the archaeological research. Archaeobotany is an essential discipline that is used to answer questions about the reconstruction of the paleoenvironment, its evolution through time, the human-environment interactions, the exploitation of and uses of plant resources, but also to answer questions about the landscapes, the impact of climate and human activities on the landscapes, the domestication of plants, plant introductions in new geographical areas, the development of agriculture, the subsistence economies, the ancient trade across the Mediterranean and also in other regions of the world, the seasonality of practices, the social organization, the human cognition, that is how humans perceive the paleoenvironment and the plant resources, but also to understand the role of plants in human societies, in human and animal diet, and also to better understand the use and function of artifacts and archaeological spaces. Archaeobotany is the study of botanical remains recovered in archaeological context and can be distinguished in two main domains, the study of macro remains and the study of micro remains. Macro remains are seeds and fruits, the wood charcoal or wood that is not preserved by charring. And on the other hand, the micro remains that are observed in the microscopical scale that include the pollen grain analysis, the phytoliths, and starch grain analysis. Carpology is the study of seeds and fruits, and it comes from the term karpos in Greek, that means fruits. Anthracology is the study of wood charcoal remains recovered in archaeological sites, and xylology is the study of wood remains that are uncharred. This is the case, for example, the study of the structure of shipwrecks. The micro remains include the analysis of pollen, that is palynology, the phytolith analysis, that are rigid microscopic structures made of silica that are formed in plant tissues, and the analysis of starch grains. And here you can see some photos of these remains. What are the steps of the archaeobotanical analysis? First of all, the observation and identification of the archaeobotanical remains. That is to observe the anatomical and morphological criteria and to attribute it to a known taxon. For example, in the first case, we can see a transversal section of a deciduous oak, and we can associate it with a modern known taxon. The second step, of course, is the quantification, that is to calculate what is the number of remains and the minimum number of individuals of these archaeobotanical remains. And of course, the third step is the interpretation of all these archaeobotanical remains to put them back in the archaeological context and associate them with other information. And of course, this is not something new. It's exactly the same process we use for any other archaeological material. As for example, in the study of ceramics, we will start first observe and identify what is the pottery, understand what is the minimum number of individuals, and then do the interpretation. Archaeobotany, of course, is an interdisciplinary approach. We use information and tools that come from different disciplines. For example, of course, botany, plant anatomy, morphology, but also biology, the evolutionary history of plants, the historical text, experimentation, iconography, ethnography, and ethnobotany are also very useful for the understanding and interpretation of archaeobotanical remains. And of course, other analyses, such as the chemical residue analysis on pottery or other artifacts. So if we look back again to the three main steps of the archaeobotanical research, we can see that botany, biology, and plant morphology and anatomy are mostly implicated in the part of observation, identification, and of course quantification, because if we know what is the plant part, we can this can help us on the quantification of archaeobotanical remains. And all the other approaches are used in the interpretation. So this is exactly the part where the reference collections are very important. The archaeobotanical material, when studied at the laboratory is observed and identified through the comparative anatomy approach, that is a comparison of the archaeobotanical remains to well-known identified modern botanical remains. We will observe the anatomical and morphological characteristics and compare it to these well-identified specimens that we will find either in the identification atlas or in the reference collections. There are many useful identification atlas or identification keys that have been published, and the reference collections are, of course, very essential to this work. The reference collections are an essential tool for the archaeobotanical study. They are composed by well-identified modern botanical specimens and that are easy to use and observe in the laboratory and also to manipulate. And what is also very important is that they are composed by many individuals. So this is one of the main differences to identification atlases, where there we usually have one or two individuals that are represented. In the reference collection, we have more individuals. And so where we can find these specimens to create the reference collections, we can find them and do sampling from herbarium collections, from seed banks, exchange seeds or wood from botanical gardens, or do sampling in wood collections. Or, of course, the other method is through the sampling in the field. Of course, the first methods have some limitations, because usually we can, if we're allowed to do sampling in herbarium or seed bank collections, we're allowed to sample 1, 2, maximum 5 or 10 specimens. And in many cases, we're not allowed to do sampling because these collections are institutional or even heritage collections. So we are not allowed to do this destructive sampling. And on the other hand, another problem is the reliability of botanical identification. For example, if you have only one sample of fruits or seeds that is not associated to herbarium specimens, the botanical identification, we cannot be sure that the botanical identification is the right one. Keep in mind that in botany, the taxonomy, that is how botany and plant family, species and subspecies are organized, can change when we do and when we develop the genetic stats, etc. So it's very important to keep a trace and make sure that we can come back and verify the identification of the samples. So after saying all this, we can understand that sampling in the field of modern botanical specimens is the most appropriate method to create the reference collections. The advantages of this method is that we can sample multiple specimens, we can go back and sample more if needed, we can associate these seed or wood samples to herbarium specimens, like the ones you can see on the right. So we can make sure the reliability of this identification. And of course, we can collect many other information about the ecological conditions, the habitat, the phytosociology, the soil where these plants grow up, etc. So now I'll go next to the next part, and I will talk about the constitution of this reference collection during my PhD studies from the field to the laboratory. So this material was created through multiple field missions that were organized in various sites in Cyprus. Here you can see a map with the different localities that we have visited multiple times to do the sampling. Of course, the sampling was in respect to the legal framework of the Republic of Cyprus, the European Union, and the international conventions, such as the Phytosanitary Pass or the Nagoya Protocol. And as you can see, we visited multiple times the same localities in order to do the collection of different plant parts, that is, leaves, flowers, to create the herbarium specimens, wood to create the anthropological collection, and of course seeds and fruits. And as you can understand, there's a cycle of life of the plants in 1 year. So the herbarium specimens were collected during the spring or early summer. The wood were preferably collected in in the winter, and of course the seeds and fruits in late summer and in autumn. We visited more than 30 localities in Cyprus, covering almost all the possible altitudes. And why was it important to cover all these altitudes? Because the vegetation is strictly connected to the altitudes and, of course, the climatic conditions. So this approach permitted us to sample plant material from as many as possible different vegetation types, such as the frigana or maquis that are tested at the lower parts the lower altitudes of Cyprus, the deciduous and pine forests, even at the highest mountains of Troodos. And of course, we also included the plants that grow in other vegetation types, such as in the riparian forests. Here you can see an example from the riparian forest near Hierokitia, or even and also the Halophilus vegetation that grow near the salt lakes of Larnaca. There was a very particular method that we followed and a very particular protocol that we followed during the field missions. We created this form, where we noted in English all the information about the sampling. The first part was dedicated to the date and sample locality. The next part was dedicated to the sample description, if it's a branch, leaves, if there are flowers, wood, or fruits. From which part of the plant we did the sampling. And the next part was dedicated to the taxonomic identification, starting from family, species, subspecies, if there is a subspecies. And we also noted the common name that is very useful when we do the ethnobotanical studies as well. The last part was dedicated to the information about the habitat, the ecological and phytosociological information of the sample. And of course, we took photos of the plant and also of the environment. One very important part in this documentation sheet is the code. There was one reference collection number that was given to each sample, independent of the family or the species. And this is the reference we use through the reference collection, and is preserved at all the processing of the reference collections, and it's even preserved when we did the conditioning of the specimens. For example, at this photo you can see the final product of one sample of the anthropological reference collection, where you can see the name of the family, of the species, and in bold the reference collection number. All this information were then treated in an Excel database that has been deposited at the 3 institutions where these collections have been given. I will talk about it a bit later. The processing of modern botanical samples. Let's start from the anthropological collection. The wood was dried in the open air or using an oven, and this was done with the help of Michel Lemoine at the Natural History Museum in Paris. Again, we followed a very precise protocol for the creation and the conditioning of the reference collection. In order to ensure that the wood is very well dried, we did the weighting of the samples every some days, and when this was stabilized, we considered that they were dried. And then they're carbonized in the oven for 400 degrees Celsius for 1.5 hours. And then once they were carbonized, they were conditioned in plastic boxes, using again the label with all the important information of the samples. After doing that, all the samples were observed at the laboratory under a metallographic microscope in order to do a description of the wood anatomy and also take photos using the scanning electron microscopy. This was done in order to create an identification atlas where, for example, you can see at the right hand, we have all the information about the sampling, the distribution of the plant species, and a detailed description about each species together with the photos. Coming to the carbological collection, that is a collection of seeds and fruits. The specimens were put in the refrigerator for 1 month, and we did this in order to kill all the insects that they can destroy your reference collection. And once they were ready, we did manual cleaning in order to separate the fruits and the seeds and other plant parts that we didn't wish to keep. In some cases, we purchased some seeds, for example, for some introduced species that are not indigenous on the flora of Cyprus. When we were doing the cleaning of the fruits and the seeds, we had in mind to keep other parts of the fruit or the plant that sometimes can be found in the archaeological context and can be useful to be kept in the reference collection. This is, for example, the case that you can see on the right. This is poppy seed. We kept the poppy seeds in the tubes, and also we kept the capsules that are important for the identification. And finally, for the herbarium collection, the branches with bearing the leaves or the fruits when it was possible, they were first cleaned manually, and then they were dried in newspapers for 3 to 4 months. We followed the instructions given by the Department of Forests and our colleagues, to whom I'm very grateful. And once these specimens were well dried, they were put on white paper, in order to create the herbarium specimens. And as you can see on the right, we included all the information about the specimen, including the reference collection number. The taxonomic identification was done either in the field during the sampling, through the observation of the morphology of the plant, or if it was not possible to do it up to the species or subspecies level, we did a verification of these identifications using the herbarium specimens we created, and also the photos, and of course using information coming from the flora and other botanical description of the plants. Again, I'm very grateful to Charalambos Christodoulou and Angelos Kirakis, two specialists of the flora of Cyprus, who contributed a lot to the verification of certain identifications, and they work at the Department of Forests and at the Agricultural Research Institute of Cyprus, where these collections were also deposited. So, what's the importance and scientific value of these reference collections? These collections, this work conducted to the creation of one of the richest reference collections of the flora of Cyprus that are present in Cyprus and also abroad, and they are also very representative of the Mediterranean flora in general. As you can see on the first table, more than 200 specimens were created, including a very large number of families, genera, species, and some subspecies. Especially for the anthropological collection, we consider that we have that in our reference collection, that our reference collection represents more than the 80% of the indigenous flora. These are the first carbological and anthropological reference collections created at the University of Cyprus that will be used for the archaeobotanical analysis. And another point of value of these reference collections is the reliability of the taxonomic identification, not only because we did the maximum to identify, to do the best taxonomic identification during the sampling or after, but especially because all these samples are associated to herbarium specimens. And so we can come back at any time and verify this taxonomic identification. Another point to highlight the importance of these collections is the sustainable preservation and development of these reference collections, that are the different duplicates that have been deposited in different institutions in Cyprus and abroad, and of course the stock of dry wood that will help in the sustainable preservation of the anthropological collection. In the second table, you can see the duplicates deposited in Paris at the National History Museum, at the University of Cyprus, of course, and a copy of the herbarium collection is deposited at the Department of Forest. A third copy is included in my personal reference collection. This helps to the enrichment and the addition of new specimens of the rich collections of the Natural History Museum in Paris, as we included new well-identified specimens and also new taxa that they did not exist before, such as several endemic species of the flora of Cyprus. These reference collections are not only important for archaeobotanical and archaeological research, they're also very important for other studies. But here I would like to say a big thank you to the French archaeological mission of Hierogidia and also at the University of Cyprus, who co-funded the purchase of a metallographic microscope that is now at the Archaeological Research Unit and can be used for the analysis of anthropological remains that we find in archaeological sites in Cyprus. This is essential for the creation of a laboratory of archaeobotanical studies at the University of Cyprus, and of course for the development of archaeobotanical studies in Cyprus and in the Mediterranean in general. And as I said before, the constitution of these reference collections, but also the funding of this microscope do not only contribute to the development of archaeobotanical studies, but they also contribute to other disciplines. For example, the paleoethnobotany, paleoecology, agronomy, ecology, botany, and the study of the modern flora of Cyprus and the Mediterranean. During our field missions, we had the opportunity to do observation about the distribution of plant species, their habitats, their ecology, and their phytosocioecology that are very important also for specialists working on the modern botany and flora of Cyprus. And of course, during all this process and all these years, we were in very close collaboration with other specialists of the flora of Cyprus, and we provided also all the information that we collected. And this was, for example, the case of the identification and localization of new individuals of lesser known species, subspecies, or hybrids in Cyprus, as it was the case of a hybrid of Pistacia and of a near endemic species that was identified in the field and also in the archaeobotanical material as well. That is Cyprinia gracile, that you can see the photos on the bottom of the slides, and the publication was done this year in the journal Cypricola. Of course, there are many perspectives, as you can understand. We'll continue to develop these reference collections by adding new and other taxa of the Cypriot flora. We'll continue developing the identification atlas of the anthropological collection, but also of the carbological collection using drawings and photos of the specimens. It's a wish to create an online database in order to digitize all this information that will help us to disseminate this information at the scientific community, also abroad. It is also a wish to continue developing these reference collections by adding other types of micro and macro plant remains, such as phytoliths, pollen, starch grains, roots, and tubers. And of course, as we are developing also the ethnobotanical studies in Cyprus, we will wish to include some ethnobotanical specimens that maybe they don't exist in some museums. And it's very important also to include them because, as I said before, They are very helpful in the interpretation of archaeological and archaeobotanical remains. And of course, we will continue to further collaborate with other institutions in Cyprus and abroad, such as botanical gardens, natural history museums, and universities, to develop and disseminate all this work. Before closing my presentation, I would like to thank Alexandros Onassis and Siviano Foundation, who funded my PhD studies. I would like to say a big thank you to my supervisors, Urania Kukan and Margareta Tenberg, who have been by my side all these years and who have been inspiring me through their work and their support. I would like to thank the directors of the Mission Néolithisation, François Privat, Jean-Denis Vigne, and Jean Guillain, and also the directors of the French Archaeological Mission in Chirokitia, Alain Lebrun, who give me the permission to study the archaeobotanical material from their sites and give me the opportunity in this framework to develop and create all these reference collections. Of course, I would like to thank Odile Lebrun, Laurence Astruc, Françoise Lemort, for giving us the opportunity to go fund, along with the University of Cyprus, the microscope that we purchased. A big thank you to Thanasis Vionis as well, who for his major contribution in purchasing this microscope. Michel Lemoine, who contributed to the creation of the reference collection and their preparation at the Natural History Museum in Paris. Charalambos Christodoulou, Angelos Kiragis, and the directors of the Department of Forests and the Department of Environment and Agricultural Research Institute for giving me the opportunity to visit their localities and to have this very close collaboration all these years. And of course, a big thank you to my parents, with whom we visited all these localities in Cyprus, from the lowest altitudes up to the highest mountains of Rhodos, and other people who gave me the permission to do the sampling in their private vans and to include and create this reference collection that I presented to you today. This presentation is dedicated to all of you. Thank you.
Well, thank you, Maria, very much for the presentation this evening, but above all, for creating this collection and for donating this to the Archaeological Research Unit, a renowned institution in the Eastern Mediterranean. As you can all see, a former student or former PhD student of the University of Cyprus became, in case of Maria, a sponsor for further studies. Maria has already thanked all people who have contributed to the creation of this collection. From my side, I would like to thank very much my colleague from the National Museum of Natural History, Margareta Tenberg. Hello, Margareta. Thank you very much. And of course, Maria's parents for the crucial contribution to the creation of this collection. I suppose that many of you who are joining with us in the audience or who are attending us from far away, you are having questions to Maria. She's ready to answer them.